Welcome back students to this section of MCQs. So let us start with the first question. Which of the following coenzyme forms of vitamin is required for the synthesis of catecholamine neurotransmitters? So your answer should have been pyridoxal phosphate. Now please remember that pyridoxal phosphate is required by the enzyme dopa decarboxylase. All the decarboxylases actually require pyridoxal phosphate. Tyrosine is converted to tyramine, dopa is converted to dopamine, histidine is converted to histamine. All this formation requires pyridoxal phosphate where there is decarboxylation, and these are the ones which are formed are called as biogenic amines. Now out of the biogenic amines, some of them are catecholamines. Catecholamines actually involve three catecholamines: dopamine, adrenaline, noradrenaline. These three form the catechol. They have a catechol nucleus and thereby they are called as amines, catecholamines. Now for the synthesis of dopamine, pyridoxal phosphate is required, but for the further synthesis it is from dopamine itself norepinephrine is synthesized, and from norepinephrine epinephrine has to be synthesized. There is no other pathway for the synthesis of norepinephrine or epinephrine. The only way they can be synthesized is from dopamine. So from the either in the CNS or in the peripheral nervous system, everywhere whenever adrenaline is going to be synthesized, it has to come from dopamine or from norepinephrine to epinephrine. So this is the pathway. So for the formation of dopamine, coenzyme is required, dopa decarboxylase, and that requires pyridoxal phosphate. I have already told you thymine pyrophosphate is mainly involved in carbohydrate metabolism. NADPH. Now NADPH is required in HMP shunt where it is produced, and it is required in only the de novo synthesis, reductive biosynthesis, anabolic pathways, like maybe the synthesis of cholesterol, synthesis of fatty acids. In all these, where there is reductive biosynthesis, NADPH is required. FMN is only in two reactions within the body. One is the L-amino acid oxidase and the other is electron transport chain, NADH dehydrogenase. Tetrahydrofolic acid is mainly involved in one carbon metabolisms, and so it is not required for the synthesis of catecholamines as such. S-adenosyl methionine is required, but tetrahydrofolate as such is not required for the synthesis of catecholamines. But directly it is not required. So the enzyme correct answer could be pyridoxal phosphate. Let's go to the next question. The folic acid derivative used in the synthesis of thymidylate, formyl tetrahydrofolate, form iminotetrahydrofolate, methyl tetrahydrofolate, methylene tetrahydrofolate, methinyl tetrahydrofolate. Very confusing options. So what was your answer? The answer should be methylene tetrahydrofolate. Methylene tetrahydrofolate is the one which acts as a coenzyme for thymidylate synthase. How is this formed? It is converting. Remember, it is DUMP is converted to TMP, deoxythymidine monophosphate actually. Now for this, in this it is extra methylation that is taking place of UMP to form of the uridine base to thymine base. We require an extra methyl group, and this methyl group comes from methylene tetrahydrofolate. Methylene tetrahydrofolate. Now one characteristic feature of this reaction is that methylene tetrahydrofolate donates its group and it is converted to dihydrofolic acid. So this is one characteristic feature. Methylene is converted to dihydrofolic acid. Remember, it is only UMP, the monophosphates ones, which can be acted upon by methylene tetrahydrofolate. And this is formed, this has come from deoxyuridine diphosphate, and deoxy can become from uridine diphosphate to the deoxy form ribonucleotide reductase. Ribonucleotide reductase cannot act directly on UMP and convert it to deoxy UMP. It has to be UDP or the diphosphate. Only then ribonucleotide reductase can act on it, convert it to the deoxy form. Deoxy form is further again remove the phosphate group, convert to deoxy UMP and then to TMP. What's the difference? Remember, in thymine monophosphate, thymidylate monophosphate, what is required is that this has to be, the ribose has to be deoxy sugar, because T is involved only in DNA synthesis and not in RNA synthesis. So in DNA, it is always deoxyribose. So this has to be deoxy form only. DUMP is the one which has to be converted to TMP. It has to be, the sugar has to be in the deoxy form, since we are talking of RNA and DNA. This will be in the, will be present, the UDP and all will be present in RNA, and this will be present in DNA. And the enzyme, folic acid derivative is methylene tetrahydrofolate, which is converted to dihydrofolate. Let's go further to the next question. The functions of vitamin C include all of the following except. So did you get your answer? The answer is vitamin B12 absorption. So it is involved in collagen biosynthesis. Yes, that's why in scurvy we have got loose gums, bleeding gums, etc. Why? Because it is mainly involved in collagen formation. How is it involved? Lysyl hydroxylase and prolyl hydroxylase are responsible for the hydroxylation of the lysine and the proline residues of collagen, and for this we require vitamin C. So in vitamin C deficiency, there will be loose gums, bleeding gum, teeth, etc. will be there because of the lack of collagen biosynthesis. Adrenaline synthesis. So where did it come? It is a catecholamine. So from where did it come? So we have got tyrosine hydroxylase to dopa to dopamine. So in this pathway also vitamin C is required. Phenylalanine to tyrosine to dopa to dopamine. In this pathway also vitamin C is required, and thereby vitamin C is also involved in adrenaline synthesis. Bile acid synthesis, yes, again 7-alpha hydroxylase, the enzyme which converts cholesterol to the bile acids, cholic acid or chenodeoxycholic acid. The whole conversion of cholesterol to bile acids is taking place in the liver, and this formation of bile acids, it is a lot of steps are involved, but the regulatory enzyme is 7-alpha hydroxylase, and 7-alpha hydroxylase, or in further at least, will require vitamin C. So for the formation of cholesterol to bile acids, we require vitamin C. Then it acts as an antioxidant. Yes, it is the one of the cytosolic antioxidants, or water antioxidant of the water form. Vitamin E is mainly a lipid antioxidant, whereas the aqueous antioxidant is vitamin C. Vitamin C is also reacts with vitamin E and thereby regenerates the vitamin E more and more by action. It forms as an link between the lipid antioxidants and the aqueous antioxidants. The connection is between vitamin C and vitamin E, and so it acts as an antioxidant. So what about vitamin B12 absorption? B12 absorption is very specific, and it requires a glycoprotein called as intrinsic factor or intrinsic factor of Castle, and B12 is actually called as extrinsic factor of Castle. So this intrinsic factor and extrinsic factor will combine, and only then B12 will not be degraded in the stomach. Otherwise B12 would have been completely destroyed in the stomach. To prevent its degradation not only in the stomach or further down in the duodenum or anywhere, it has to associate with intrinsic factor, and it is the intrinsic factor B12 combination. The complex is the one which is going to be taken up by the different enterocytes. So it is in this, vitamin C has got no role. So vitamin B12 absorption does not require vitamin C. Is vitamin C required for any other absorption? Yes, it is required for the absorption of iron. Iron is always absorbed in the ferrous form. So to convert Fe plus 3 to the Fe plus 2 form, this is the one which can be absorbed. We cannot absorb Fe plus 3. So for this conversion, we require vitamin C. So higher the vitamin C content within the food, more is the iron absorption, because vitamin C will convert ferric to ferrous. So that is where it is required in the absorption, but not in vitamin B12. Its main function in the GI is in over here, conversion, iron absorption. So let's go to the next question. The functions of vitamin A include all of the following except. So it should have been easy for you. Except coenzyme for fatty acid synthase complex. Vitamin A is a sort of like, it acts in the same as that of a steroid hormone. So vitamin A, especially the retinoic acid form, it has three forms: retinol, and retinoic acid. Now the retinoic acid is the one which is responsible for the different other functions that you are seeing here: glycoprotein synthesis, growth and differentiation. All this is possible because of retinoic acid, and retinoic acid acts similar to that of a steroid hormone. So what it happens is, remember, these are fat soluble vitamins, so it will cross the cell membrane. Once vitamin E crosses, retinoic acid crosses the cell membrane, it binds to its specific receptor. Now, minute it binds to its receptor, this receptor will undergo a change. It will undergo a conformational change. It undergoes a conformational change. Now, this receptor can go cross into the nucleus, and it can go and bind to the DNA at specific sites, and it will go and bind to the DNA on specific sites. It will come and bind, and thereby it will regulate gene expression. So retinoic acid will go and bind to specific areas on the DNA, and these areas are called as it's like a response element. So we call it as retinoic acid response element, and thereby it regulates gene expression, formation of mRNA, and finally formation of the different proteins. Whatever proteins have to be formed are all regulated. So retinoic acid has a role in growth and differentiation. Now what about retinol and retinol? Retinol is mainly with reproductive system, and retinol is mainly with vision. Levensis retinol. Opsin plus levensis retinol is rhodopsin, and rhodopsin is responsible for the dim light vision which is possible in the rods. Glycoprotein synthesis, again it is by this mechanism glycoproteins are synthesized, retinoic acid. And growth and differentiation, again because of this mechanism. And some retinol also may be involved in reproductive system and its growth and differentiation. Antioxidant, yes, vitamin A is a very good antioxidant. Especially beta carotene is considered to be having more antioxidant activity. Vitamin A, pro-vitamin A is beta carotene. Beta carotene can form vitamin A, but it is also considered as one of the retinoids or carotenoids. There are two different compounds. Carotenoids, all of them have got antioxidant activity. So let's go to the next question. All of the following are true regarding the functions of calcitriol except, so your answer could have been increases excretion of phosphorus by renal tubules. Remember, it is PTH which is phosphaturic. P for P, so phosphaturic means it increases excretion of phosphate. Only by PTH increases the excretion of phosphate. So it is phosphaturic. Now if you look at calcitriol has got actions on three organs, that is bone, intestine and kidney. In the bone, what it does, calcitriol causes, depending upon the serum calcium level, it has either mineralization or demineralization. So calcitriol acts on the bone, and it causes, if the calcium levels are adequate, it causes bone mineralization. Okay, if the calcium levels are inadequate, only then it will cause bone demineralization, and that time it is acting via PTH. It can also act on the intestine. Its main action is on the intestine. What it does, it increases calcium absorption. Whatever calcium is there in the diet, it has to be absorbed. It requires vitamin D, and it also has an action on the kidneys. In the kidneys, what does it do? It increases the excretion of calcium, it increases the reabsorption of both calcium and phosphate. Only PTH increases the reabsorption of calcium. So from here also calcium is taken in. Both calcium and phosphate are reabsorbed from the kidneys. Reabsorption. All this will help to see that the calcium levels are maintained. Basically, it is going to increase the calcium levels, increase calcium reabsorption, increase calcium absorption, and also cause bone mineralization if calcium levels are adequate, or demineralization, and thereby maintain the calcium levels. by bone demineralization also, depending on the calcium level. So basically it causes calcium and phosphate reabsorption. So calcitriol increases absorption of calcium, yes. Mineralization of bone, yes. Reabsorption of calcium by renal tubules, yes. Increases excretion of phosphorus, no. It cannot, because it reabsorbs both calcium and phosphate. Calcitriol also is responsible for differentiation of the epidermal cells. Yes, vitamin D has been implicated more and more, not only in calcium metabolism, but it is supposed to act same as that of retinoic acid, what I told you, hormone response element. So there is supposed to be, it also acts like this. It is also fat soluble, so it crosses the membrane, goes to the cytosol, goes to the nucleus, binds to its receptor undergoes a change, goes and binds to the DNA and brings about changes in the gene expression. One is it increases calpindin, that you have already seen how it increases absorption by increasing the formation of calpindin, but it is supposed to have an effect on different other proteins also, and thereby it is involved in the differentiation of epidermal cells also. So let's go to the next question. Prothrombin time is used to assess the deficiency of, what is prothrombin? It is factor 2, 2, 7, 9, 10. The number should ring a bell in you, and that is vitamin K. Vitamin K deficiency can lead to changes, alteration in the prothrombin time. Vitamin K is involved in the gamma carboxylation of the glutamate residues. It is responsible for converting glutamate to gamma carboxyglutamate, what we call as GAL. Once it forms gamma carboxyglutamate, it can cross link with calcium and help in blood clotting. So vitamin K deficiency, these clotting factors 2, 7, 9, 10 are not getting gamma carboxylated. There is no post-translational modification of these clotting factors. So it will lead to increase in the prothrombin time if vitamin K is deficient. Let's go to the next question. Which of the following vitamins has hormone response elements and regulates gene expression? I think this is something related to the previous one. So out of all this, which of this? It is vitamin A. B12 is a water-soluble vitamin. So it mainly acts as a coenzyme. Adenosyl B12, methyl B12, both of them act as coenzyme. Folic acid is again a water-soluble vitamin. So that also acts as a coenzyme. So different enzymes will have tetrahydrofolate or dihydrofolate, one carbon metabolism. Vitamin A, as I said, retinoic acid is the one which is responsible for regulating the gene expression. Thiamine, again it is TPP, coenzyme. Vitamin K is also mainly, even though it is a fat-soluble vitamin, it also has a coenzyme form, and thereby it is responsible for gamma carboxylation. So for this, vitamin K required is mainly as a coenzyme form and not as a hormone response element, or does not regulate gene expression. So vitamin K causes post-translational modification of 2, 7, 9, 10. Let's go to the next question. Starvation is characterized by, so your answer should have been gluconeogenesis in liver. So rest all are false. In starvation, glucose levels are low, so insulin levels cannot be high. So it will be characterized by low insulin levels. Low glucagon levels, no. When glucose levels are low, glucagon is going to be released from the alpha cell, so there will be high glucagon levels. Low acetoacetate levels. Acetoacetate are ketone bodies, and ketone bodies are synthesized during starvation. So their synthesis will increase and their levels will be higher. Lipogenesis in adipose tissue. Lipogenesis? No, lipolysis. Hormone sensitive lipase is responsible for lipolysis during starvation. When starvation is there, why will anybody want to synthesize adipose tissue? Adipose tissue is going to be broken down. So lipogenesis cannot take place. Gluconeogenesis, yes. Gluconeogenesis is the mechanism for maintaining the blood glucose during starvation. Gluconeogenesis, the substance mainly come from muscle breakdown, and the proteins in the amino acid give the substrates, and gluconeogenesis takes place in the liver. Next question. Enzymes active in the fasting state. Again, we are looking at in fasting state include all of the following except. So based on the previous question, you should be able to tell which of this is happening. In the fasting state, what is more? Glucagon is more. If glucagon is more, it causes covalent modification. Of what? Of different enzymes. What does it cause? It causes phosphorylation. So enzymes which are active in the phosphorylated state will be the ones which are needed, which will be activated by glucagon. So look at all of these enzymes and you will be able to tell the enzyme is active in the fasting state. Include glycogen phosphorylase. Glycogen phosphorylase. Hormone sensitive lipase? No. Pyruvate carboxylase de novo synthesis of fatty acids, so not taking place. Glucose 6-phosphate is actually a gluconeogenic enzyme, so maybe not active, but the most important one among all this will be glycogen phosphorylase. So it includes all except, so glycogen phosphorylase, what is happening? Glycogenolysis is taking place. So include all except glycogen phosphorylase. Let's go to the next question. Metabolic pathway active in mature erythrocytes. Metabolic pathway which is active in mature erythrocyte. We are talking of a mature erythrocyte. So your answer should be anaerobic glycolysis. So mature erythrocyte, citric acid cycle, note mitochondria, beta oxidation, mitochondria, gluconeogenesis, liver. Ketogenesis is happening in the liver. So it does not take place in the erythrocyte. The only mechanism by which erythrocytes get their ATP is by anaerobic glycolysis. So that's the only way by which anaerobic glycolysis, pyruvate is converted to lactate. That is what happens within the erythrocyte, whether there is enough oxygen or there is not enough oxygen. The end product of glycolysis in erythrocytes, especially mature erythrocytes, is always going to be lactate. Why? Because the RBCs do not contain mitochondria. So the pyruvate cannot enter anywhere and get converted to its acetyl-CoA or enter the TCA cycle. No mitochondria. Therefore, always, whether it is anaerobic condition or aerobic condition, in a RBC, it is always anaerobic glycolysis, and this is the source of ATP for RBC. So with this, we come to the end of this section of MCQs. Let us do it further in the next section. Thank you.